This is lecture 33 in the speciation section of ZOO3649, Evolutionary Genetics. And today, so the last two lectures we've talked about the classical view of speciation, okay? Dobzhansky's view of tiny little incremental changes that eventually lead to speciation, or evolution of different species. And the classical view was determined by geography. But the main defining feature of the classical view was that gene flow had to stop in order for speciation to happen. Okay? That is the defining view of the classical way in which we thought of speciation. That gene flow has to stop. Okay? Either gene flow stops because there's a barrier or there's non-random mating. Okay? So, one way or the other, gene flow has to stop for the classical view to lead to speciation. All right. So, in these two lectures, I'm going to talk to you now about the modern view of speciation, which is becoming more and more and more clear the more research gets done in the world. Okay? And especially now that we have genomes, not just little pieces of mitochondrial DNA or microsatellites, we have whole genomes and bioinformatics. And so we can analyze big pieces of ancestry and work out things that we were not able to work out before. And so that leads us to our modern view of speciation. Okay? So, and again, guys, this is important enough for me to be stressing. You will be examined on this. There will be exam questions, not one, many, okay, on these aspects. And you better know it, okay? If you don't know it, please watch the lectures again and again and again. This stuff is all going to be examined. Okay, so the modern view, it stems from the start of this modern idea, stems from a publication by the Marie Curie Speciation Network, this European network, where asked, what do we really need to know about speciation? And the focus of this modern views moves away from the geography focus. Okay, it moves away from the classical focus, which is geography and stopping gene flow, right? It moves away from this and it moves towards, its focuses on reproductive isolation, how it evolves, the genetics of speciation, okay, how allele frequencies change through time, and the patterns of species diversity we see across the world. So we are throwing away the old way of thinking in terms of geography and gene flow and subspecies. We're throwing those concepts into the dirt bin. Because why? No, I'm not saying that they are bad concepts. I'm saying that we have now become cleverer. We have now more data. And so we can see that although those concepts of the classical view of Meyer and Dobzhansky, they were useful at the time. But now we have more information. Now we can see that that classical view is not actually the whole picture. It tells us a part of the story, but not the whole story of how species are formed. And that's why we have these two lectures here, so that I can inform you, I can teach you about how we have our view of speciation has been changed in the last, say, 5, 7, 8 years since the genomic era has started. What new insights can we get? And how is that questioned the classical view and modified the classical view? Okay? So the first change is that now when we talk about evolution, we talk about populations. Okay? We don't talk about subspecies anymore. We left taxonomy behind and we focus on populations instead of subspecies. And remember, subspecies was the brainchild of Ernst Mayr. Okay, so he introduced, but when he introduced subspecies, he also knew that the concept was not a perfect concept. Okay, and the problem that comes with defining species was even worse with defining subspecies. So Ernst Mayr knew, even though he introduced this idea of subspecies, deep down he knew that this was not it was far from a perfect idea. Okay, so the idea of, but unfortunately, the idea of subspecies is remains until this day, and many biologists don't even know about this modern view that I'm teaching you about today. Okay, they still are talking about subspecies. Okay, the modern view replaces the idea of subspecies with the idea of a population. And why is this important? Because populations are what evolves, right? Populations can change in allele frequency from one generation to the next, and therefore we know it is evolving, right? We don't know if a subspecies is evolving. We don't know if a subspecies changes its allele frequencies from one generation to the next. We don't know what the effect of the four forces of evolution, drift, selection, so on. We don't know what the forces of evolution are doing to subspecies, but we know what they are doing to populations. So this is why we are now replacing subspecies with populations, because populations are dynamic and they are not static. Okay, they're not fixed like subspecies are. They allow the forces of evolution to influence them. Subspecies does not take any force of evolution into account. Okay, populations do. Okay, and this is all part of the whole idea of also moving into the coalescent, taking populations, merging them with trees, and forming the new science of coalescence. So all of this leads to this new modern approach to evolution, okay, that we are doing. And populations are dynamic, right? So all the forces of evolution can operate in that population and change the allele frequencies. Another big distinction between the classical and the modern view of speciation is this nonsense of microevolution and macroevolution. Okay? That nonsense, we also throw it away. We put it into our high school notebooks and we say, that was important to learn the ideas of populations and trees. But now, we don't need that idea anymore, because there's no micro and macro, it's all one thing. Okay, so that's the other thing that gets thrown out. This idea of the nonsense idea of micro and macro evolution. Throw it away. We don't need it, because the modern view is dealing only with those three things that I told you about. Okay, we're dealing with how reproductive isolation is how reproductive isolation evolves, the genetics underpinning phenotype, and the patterns of diversity. Okay, so that brings us to the question, is there a role for gene flow in speciation? Okay, remember, why am I asking this question? It's because the classical view says there is no role for gene flow in speciation, right? If you have gene flow between species, the fitness will be low. The hybrid will not be able to have kids of their own. So the hybrid won't continue, right? So no gene flow, right? So if you have gene flow, the two populations become similar. And so no species can be formed. That is the classical view, right? So the classical view says, and very clearly, there is no role for gene flow in the speciation process. Okay? As soon as you get gene flow, species can't evolve. Okay? That is Mayer's and Dobzhansky's classical view of how species evolves. Whether it's allopatric, peripatric, sympatric, or parapatric, all four of them involve what? No gene flow. Either there's a barrier or there's non-random mating. But the end of the day, no gene flow. That is how you get species, according to Mayer and Dobzhansky, the classical view. So, what we ask in the modern view is there a role? Are they right? Were those guys right when they said there's no role for gene flow in the speciation process? Were they right? Or were they wrong? Can gene flow create a new species? Can you have species 1 and species 2 with incomplete reproductive isolation? So they're still able to mate. Can species 1 mating with species 2 create species 3? That question, if you asked Ernst Mayr, he's dead now, he died in 1990s. But if you asked Ernst Mayr whether you could do that, whether you could take one species of animal and another species of animal, mate them and create a third species of animal, he would have said absolutely not. And remember, Ernst Mayr was one of the leading evolutionary biologists of his time. That's how much our brains and our thoughts have changed since the days of Ernst Mayr. Now we can ask without fear, can gene flow create a new species? And the answer is yes, it can. And that process is called hybrid speciation. Okay? In plants, even Ernst Mayr would say, in plants, it can happen. Okay? Because hybridization between two species can easily lead to a third species in plants. Okay? Because plants take a long time to become reproductively isolated. Hybrid speciation is actually quite common in plants. In fact, the wheat that we use, okay, the wheat that we make our modern day bread with, that wheat is a hybrid. It's not a species of its own, it's a third species. It is a hybrid between two different kinds of wheat. Okay? If you made bread with the old kinds of wheat, it wouldn't be the same as the bread we have today. Because the bread we have today is our modern wheat. Okay? And it's a hybrid between two different species. Okay, so plants can do it because many plants are what? They're polyploid. Okay? And so they can actually manage when the number of the sets of chromosomes, can actually double. They can quadruple. They can become different multiples. Plants don't have a problem with having extra chromosomes, extra sets of chromosomes. Okay? So because they don't have a problem with that, hybrid speciation has been happening since time immemorial in plants. Okay? So there is a role for gene flow. There's always been a role for gene flow in plants. And because there was a role for gene flow in plants, those founding fathers of evolutionary biology, like Ernst Mayr and Dobzhansky, should have realized that they cannot have a rule like, to get species, you need to stop gene flow. Because that rule, they thought, and even today, even in those days, only applied to animals, not to plants. Okay? Because in animals, hybrid speciation has always been very, very rare. Very few cases have ever been documented. Why? Because animals can't handle changes in chromosome number like plants can. Animals can't do polyploidy. Okay? We have all kinds of problems when we get polyploidy. You know, Down syndrome is a disease, right? And that's because why? That's because we have an extra copy of chromosome 21. Now you see the problems, just one extra, and it's only chromosome 21, not all the chromosomes, right? So in animals, just having one extra chromosome can lead to a big problem, okay? So hybrid speciation cannot use polyploidy in animals. So it has to be homoploid. It has to be segregating and then two segregating alleles coming together in a diploid organism. Okay, so that means homoploid without any change in chromosome number. And that makes it very difficult for animals to speciate, thanks to using hybrid speciation. In other words, when species 1 mates with species 2 to produce a species 3. Plants can do it. Animals do it very rarely. Okay? And Ernst Mayr believed that this hybridization between animals, species, has little or no role to play in speciation. Okay? In other words, gene flow has no role in speciation. Because why? The fitness of the hybrid phenotype is always going to be less than the fitness of the parents. like in that case with the liger, the cross between the lion and the tiger. You saw the fitness of that animal is not very high. Okay? And those kinds of cases led Ernst Mayr to think, in animals, we can't have this. Because in animals, the offspring's fitness is always going to be low, okay, compared to the parents. Therefore, those offspring will never become another species by themselves because their fitness is low, okay? So there are very, very few examples of hybrid speciation. One example, which we discovered in 2019, so very recently, and using high-end genetic data. It was a study that was published by myself and colleagues from Germany and other parts of the world, New Zealand, Australia, and so on, France. And this was a study that we carried out on a bird species called the prion, okay? And the Selvens prion looks like this. And on top of that, not only, where is my mouse? Let me try and find it. Sorry, here it is. Not only is the Selvens prion a hybrid species, okay, it is a South African species. So not only is one of the rare cases of animal speciation documented, but it's actually documented by South African scientists on a South African species, okay? So this species, Selvens prion, it has a medium width bill, okay? Its parent species, one had a wide bill and one had a narrow bill. Selvens prion has a medium bill. It's not narrow and it's not wide, okay? Because it's a hybrid between the two species. So this is, so you'll be happy to know that even research from UniVen itself, yeah, is adding to our view of biology, okay? And this paper of ours about the Selvens prion has shown very clearly that Ernst Mayr could not be right, okay? Species 1 mating with species 2 can lead to species 3. And Selvens prion is an example of species 3, okay? And so this study that we did, plus other studies in the last 5 years, have actually found a new role for gene flow and selection in the speciation process, okay? So Ernst Mayr's and Dobzhansky's view, the classical view of speciation, is already crumbling, okay? Thanks to the studies we are publishing in the modern days, okay? Thanks to our genomes and our new insights that we can get from high-end molecular genetic research. Already we are dismantling, bringing down the view, the classical view of the Ernst Mayr, or the Mayerian view, as it is called, okay? So in all the cases of hybrid speciation, one thing is common, okay? That is, gene flow occurs during an adaptive radiation. It's basically when there is the rapid evolution of one species from one common ancestor. There's a rapid evolution of many different closely related species over a very few million years, not very long. Okay? And so that rapid evolution means that each species, it's not long enough for the reproductive isolation to develop. In other words, each one is able to mate with the other one. All right? So this adaptive introgression, okay, that leads us to the idea of adaptive introgression. Introgression simply means gene flow, okay? When you have genetic exchange and the genes are passed on, is called introgression, when genes from one population enter another population. That's what's introgression. So it is another way of saying gene flow, okay? So adaptive tells you about what force of evolution. Introgression is talking about gene flow. Adaptive, if you're adapted to an environment, what happens? Your fitness increases, right? Because if you're adapted to that environment, you have a high fitness. That means you are suited to that environment, right? You're adapted to it. That means you will have a high fitness, your offspring will survive, and your offspring will reproduce themselves. So that is what means by adaptive. So it's dealing with fitness. So what force of evolution is that? Yeah, the clever ones have already guessed it. It's selection, right? So actually, adaptive introgression brings the idea of selection with the adaptive part and gene flow with the introgression part. So it's actually what adaptive introgression means is that when genes from one species come into the gene pool of another species, they give an advantage to the hybrids, okay, over either of the parents. Suddenly, it can lead to a third species because that third species, it has an advantage. What did Maya say? That third species can never have an advantage because the fitness is always low, right? That's what Maya said. But now we are saying, in some rare cases, like in Selvin's prion, species 1 can mate with species 2. And species 3, the hybrid, has a higher fitness. Not a lower fitness the way Ernst Mayr predicted. They actually have a higher fitness. And that is what is meant by adaptive introgression. Okay? So another example which came out is with butterflies, okay, in South America. And what this shows you, and here you have a phylogenetic tree, okay, a gene genealogy, all right? And in this gene genealogy, you have these two sister taxa. They look different. They look different, okay? So what is happening in these taxa? These two should be very closely related, right? Because they are sister taxa. Here they are. They are forming a monophyletic group by themselves. And yet what? They look differently. See, the pattern is not the same pattern here, right? The wing pattern of this butterfly and this butterfly, they're different, aren't they? Right. So how did they become different? Let me tell you. This one here, where my mouse is, this one here, when a bird sees this one, it has no problem to eat that one. Why? Because this one here is delicious. It's delicious. Birds love to eat them. But this one here, notice I'm not talking about this one. I'm talking about the third one now. This one here, when a bird sees this pattern here, this white pattern and that red pattern there, when the bird sees that pattern, they don't eat it. Why? Why? Because it's poisonous. Okay? The birds do not eat this one. H. timerata is not eaten. But H. melpomene is eaten because it's delicious. Okay? So, what these guys have discovered is that these two species, Melpomene and Melpomene, in fact, they are the same species. There's just a different populations. Melpomene amaryllis looks different to Melpomene agalope. Why? Why? Because this one here, it is also delicious. But what is happening with this one here? The birds are not eating them, despite being delicious. Why? Because this one here mated with this one here, and it got the color of the poisonous. Meanwhile, it's not poisonous, it's delicious. It's delicious the same as its sister. Remember, these two are very close to, they're the same species. They are both delicious. But this one did gene flow with this species, Temerata, and obtained that pattern. And once it has obtained that pattern, all the hybrids that have that pattern, okay? All the hybrids that have that pattern, what happened to them? They survived. They had a high fitness. Why? Because suddenly the birds are not eating them, right? So not only do you have introgression, gene flow, the genes of this Timerata has moved into the population of Amaryllis. And what has it done? It has converted this one which is delicious to a one that looks poisonous. It has conferred a huge selective advantage to Amaryllis. Okay? Just this mating between Timarata and Amaryllis here has given Amaryllis a huge selection advantage. Because now suddenly, those ones that look, the hybrids, not all the hybrids will look like this, but the ones that do, suddenly the birds are not eating them. And what is going to happen after many generations? Only that one will exist in this Amaryllis population. Okay? So the gene flow, the introgression between Timarata and Amaryllis has actually led to a increased fitness of Amaryllis. That is completely against what Ernst Mayr said, isn't it? Ernst Mayr said if you have gene flow between species like this, the hybrid will always have a low fitness like the lion and the tiger's baby, right? But in this case, Ernst Mayr was completely wrong. Because here you have gene flow between two species and the offspring has a high advantage. It has a high fitness. It doesn't have a low fitness. So selection favors that one. Because why? It is deep down, it is delicious, but it looks like it is poisonous. Because it got the color of the poisonous one through gene flow. And that is the first classical case of adaptive introgression, where you can have gene flow which has an advantage to the hybrid. Finally, we can put our classical speciation to rest. There is indeed a role for gene flow in speciation, in the speciation process. Okay? Because this means that a hybrid can be, the fitness of a hybrid can actually be greater than that of its parents. And that is what Ernst Mayr said can never happen. And that is why Ernst Mayr and the classical view always said, and Dobzhansky as well, you must stop gene flow before you get species. Now what we are seeing is sometimes you must increase gene flow to get species. That is the modern view. And when it comes to your exam questions about adaptive introgression, you better be able to explain that. Okay? Explain that to me. Get full marks. All right. And in the next lecture, we're going to be going further with this modern view, and we're going to be talking about adaptive radiations and how many species can evolve from one ancestor very quickly, and because they evolve quickly, the reproductive isolation is not developed. And what does that mean? When there's no reproductive isolation, it means that you are free to mate with each other, right? Let's talk more about that in the next lecture.